Hello, everybody. Welcome to the webinar on an Ensemble approach for optimizing RNA-Seq alignment. My name is Anoop Grewal. I'm Product Marketing Manager here at Bina. I'm one of your presenters today. Also presenting with me will be John Mew, our research engineer here at Bina Technologies. He's worked extensively on our merge tool for RNA-Seq alignment. Just a reminder, at the end of the presentation, we're going to have a short Q&A session, and if you want to ask a question, just make sure you hover over the green bar at the top of the screen, and that will open up a gray bar or a control panel with the option to click on the Q&A. Click on the Q&A to open up the chat window. And when you type in your question, be sure to send it to all panelists. That way we'll all be able to see it and can address your questions. And just to give you a little bit more about the chat box, when you select Q&A, the chat box opens up. Click My Q&A and go to the Ask a Question box. Enter your question. Make sure you've selected to send the question to all panelists, and then click Send. You can toggle between the My Q&A to All Q&A to see other questions then that are being asked. All right, let's get started. So RNA-Seq as a data analysis tool is gaining momentum. It's a means to look at gene expression and to characterize the transcriptome in multiple ways. It offers greater sensitivity in the form of the ability to detect rare transcripts, greater accuracy in terms of the fold changes one measures when comparing RNA gene expression in multiple conditions, and a broader dynamic range, that is, than microarrays, that is, you can detect low-level transcripts as well as high-abundance transcripts. What's also nice is that it's an unbiased approach for transcript detection. You don't have to devise the probes ahead of time or know the sequence of the transcripts ahead of time a priori to, in the case of microarrays, build a microarray to detect transcripts. So it's a nice unbiased approach and allows for a digital measure for assessing gene expression. Now, with lab cores and our research labs bringing on more sequencing instrumentation, with the cost per sample dropping, people have increased access to RNA-Seq, and so that's part of the reason that we're seeing greater adoption of the technique. You can also perform RNA-Seq down to the single cell level. So there's some new ways to assess cell transcriptional dynamics because of that. The first few publications in RNA-Seq came out in 2008, and that, as you can see, there's been an exponential rise since then. And in 2015, by the end, we're anticipating we'll have over 2,000 RNA-Seq publications. What are the sorts of questions we can answer using RNA-Seq? Well, the most common question is to assess gene expression between multiple different conditions, typically a baseline and some sort of affected condition. So you can compare treated to untreated, wild type to mutated, or disease to normal. And you can assess the gene expression of both rare transcripts and highly abundant transcripts. You can also take a look at the isoforms of RNA that are being expressed. So, for instance, it may be the case that in the basal condition, one particular transcript is expressed from a gene, and in the treated condition, the splicing may switch to produce an alternative transcript, and that can be assessed using RNA-Seq. There's also the ability to detect novel RNA, and then also, very interestingly, you can, if, for instance, one allele is mutated and the other is wild type, you can assess if genes are being expressed in an allele-specific manner. So, for instance, compare expression from a paternally derived allele and a maternally derived allele. Now I'm going to go over the workflow in the lab for mRNA-Seq. There is also the option to do RNA-Seq on other types of RNA, but this particular slide will focus on mRNA-Seq. Typically, you're starting, as I mentioned, with 2 or more conditions that you're looking at. In addition, if you perform biological replicates, you're going to get better estimates for fold change, more statistically significant. So in fact, you may even be doing 4 or more samples per experiment. Total RNA is extracted from each sample, and when you look at the mix of the total RNA, it's actually the ribosomal RNA that makes up over 95% of that population. The mRNA is going to be about 3%, and then there's also long non-coding RNA and short non-coding RNA. And so then to enrich for the mRNA, a poly-A selection is most often performed using oligo-DP typically. The alternative may also be to use ribodepletion to get rid of some of the ribosomal RNA. After that, RNA is fragmented, converted to cDNA, adapters are ligated, and then typically that RNA is sequenced. This can also be an intervening step where if you're looking for a specific population of RNAs, let's say for instance tyrosine kinase, you could incorporate a targeted enrichment step. And then to the analysis. So this is focused on the analytical methods that are available through our secondary analysis solution, Bina. As I mentioned, you're looking at least 2 samples representing 2 different conditions, and ideally you'll have replicates for each of those conditions. And so you'll start with the raw reads, and alignment will be performed with your choice of top hat, star, or HighSet, or all 3. And then if you're looking for novel and annotated transcripts, you would go ahead and perform transcript assembly using couplings. If you're looking also at differentially expressed transcripts and want normalized expression, then in addition to the transcript assembly by couplings, you'll perform expression quantification using either couplings or feature counts. And for quantified expression within couplings, it's cuff-diff and cuff norm that are being used. You'll also find that within the Bina solution, there's extensive quality control reporting, and you have your choice of 3 different sets of quality control metrics. Now, as I mentioned, the RNA-Seq analysis is performed using our Bina solution. There are multiple different analytical workflows available through there, and just one of those is the RNA-Seq workflow. As I mentioned already, you have the option of identifying novel and annotated transcripts and or performing expression quantification. So what does the UI look like? It's pretty straightforward. You go ahead and choose the RNA-Seq workflow and then decide which set of quality metrics you would like to generate and view. You have your choice of SAS-QC, Picard QC, and RCQC. And then for alignment, you have your choice of HighSet, top hat, and star aligners. And as I mentioned, you'll also with the merge tool be able to select all 3. For assembly and expression, if you just want to look at annotated transcripts, you can go ahead and select couplings and or feature counts to look at the differential expression. Alternatively, for novel and annotated transcripts, you can use couplings for that. Now here's an example study that highlights what insights can be gained from using RNA-Seq analysis. Daniel, Craig, and co-authors looked at metastatic triple-negative breast cancer. And this is a type of breast cancer that's less common and much less well-studied than the HER2 and the hormone-responsive breast cancers. And so the opportunity here with DNA and RNA-Seq experiments is the potential to identify potential drug targets for future therapies. Now, these authors looked at 14 different tumors from 14 patients, and here's some of their RNA-Seq findings. When they looked at the P53 gene, there were 9 tumors that had an exon-coding TP53 mutation. In 7 of those 9, more than 90% of the expression was coming from the mutated allele. So something you would not have been able to figure out just by looking at the DNA-Seq results. FOXM1, for that particular gene, no mutations were found. And yet, from the RNA-Seq analysis, 12 out of the 14 tumors were overexpressing it. FOXM1 is associated with cell proliferation, and so this is a potentially very interesting finding. With respect to the retinoblastoma gene, a splice site mutation was found, and when they looked at the RNA-Seq results, it appears that an exon from a highly conserved domain was what was missing from the transcript. Now, if we take a look at the right, that's where we can see some integration of results taking a look at both the DNA-Seq and the RNA-Seq. One would predict, for instance, if you have a DNA deletion, as you see for the P10 tumor suppressor gene, you may also see underexpression of that gene. And sure enough, for sample 11, the P10 gene is both deleted, the copy number is decreased, and there's underexpression. But in the case of samples 1 and 9, that's not what's seen. BRCA2, similar results for a particular sample. In the case of a RAF gene in one sample and a RAF gene in 2 samples, DNA amplification was observed, and in concordance with that, RNA overexpression was observed. But it's been shown before that the copy number change does not necessarily always result in a change in the over and under expression. So it's good to confirm that. And so within these pathways, in the case, for instance, of the FGFR gene, even though a DNA amplification was observed, the corresponding FGFR gene was not seen to be overexpressed. Now I'm going to hand over the presentation to my colleague, John.
Thank you, Anoop. So as Anoop alluded to, detecting aberrant splicing in RNA-Seq is very important, such as detecting novel isoforms. And to be able to detect novel isoforms, you need accurate and comprehensive spliced alignments. However, in studying different RNA alignment tools, we've discovered there is significant discrepancy between the junctions called from the various aligners. So here in this example, we aligned 200 million 100 base pair RNA-Seq reads from human embryonic stem cell with 3 different aligners, STAR, Palpad 2, and HighSet. And looking at the private junctions discovered by each of these tools, there are significant numbers of private junctions. And these are junctions which were discovered by each individual aligner, but not by the other 2 aligners. And this discrepancy may mean that if you use only one aligner for your data, you will miss certain junctions, and then these could be important to your study. So to address this, we developed an ensemble approach called RNA-Jam, which takes the alignments from many different aligners, in our case, 3 different aligners, Palpad 2, HighSet, and STAR, and then merges them to form a merged alignment STAM file. This STAM file can then be subsequently passed to transcript assembly and expression to get the isoforms and the differential expression. So our RNA-Jam essentially takes the alignments of each aligner, rescores them, and then picks the best scoring alignment. So it will pick the alignment that it feels is the most accurate in terms of comparing to the genome. So in order to understand why we choose these 3 aligners, I'll briefly go over the algorithms of each 3 and the differences between the 3 alignment tools. So Palpad is a classic split read approach to RNA-Seq alignment. It divides the read into segments and then aligns each of these segments to the genome via a standard DNA-Seq aligner. So in its case, Palpad 2. If any of these segments do not align to the genome, then it is aligned with a split read approach, which can discover intron junctions. However, the disadvantage of this approach is that if a splice junction occurs near the edge of a segment or near the edge of a read, then the split read approach may not be able to capture the intron junction. Also, it is not so tolerant to mismatches to the genome. In contrast, STAR does not rely on a genomic aligner. It builds its own DWT-based index of the entire genome, and then it aligns the read in a very natural approach. So it starts from one end of the read and extends the alignment one base at a time until it cannot extend the alignment anymore. At this point, it assumes that because it can't extend, it has hit some intron junction, and then restarts this extension process in order to find the next intron and the next intron junction, the next exon, the next intron junction, and so on. So this is a very natural way to align RNA-Seq reads, and it's very fast. It can also handle mismatches quite well, and also intron junctions anywhere in the read. However, a potential pitfall is that if there is a process pseudogene which is quite homologous to the real transcribed gene, then the extension process can extend into the pseudogene and potentially miss the actual real gene alignment. HighSet is similar to STAR in that it also builds this global index of the entire genome. However, in addition to this, it builds local indices of, I think, 64 kilobase regions covering the entire genome. The idea of this is that instead of doing the global search for the entire read, the global search is only done for a portion of the read, such as like a seed region, and then this restricts the alignment to several local indices. And then the remaining of the read is aligned using these local indices. This allows a more comprehensive search for the intron junctions within these local regions, and can potentially avoid pitfalls such as aligning into pseudogenes. However, because of these local regions are 64 kilobase in size, if there are very large intron junctions, which are much, much larger than these windows, HighSet sometimes has trouble with it. So those are the three alignment tools, and we see that each of them has their own advantages and disadvantages. So we perform a simulation study to see what merging will do in terms of improving the performance. So in this simulation study, we simulated 50 million reads with RNA-Seq simulator based on expression from real NA127 RNA-Seq data. We aligned these reads with all three aligners considered, and then because this is simulated data and we know the ground truth, we can distinguish true positives and false positives. As expected, if we take the union of all three aligners, we're able to get the most junctions overall. However, by simply considering, say, the junctions called by at least two aligners, we don't reduce the total number of junctions found too much, but we significantly reduce the false discovery rate. So this can be this trade-off, whether you're interested in being as comprehensive as possible or improving the specificity of your study. So looking at the private junctions of each tool, we see even with this very clean simulated data, there are still private junctions for each tool. And these are junctions which cannot be found by the other two tools. And this, again, highlights what we've been mentioning, that you have to look at all of the aligners to be able to capture the most comprehensive view of this transcriptome as possible. Although these private junctions have much higher FDR, there are still true junctions here, which are not captured by the other tools. So looking at some concrete examples of true junctions which are missed by other tools. So you can see a junction here where HighSet and Palpad is able to align, but is missed by STAR, possibly due to parallels. Here we have a junction which is able to be aligned by STAR, but completely missed by the other two aligners, maybe due to the number of mismatches in one end, due to sequencing errors. now we have another junction, a true junction, which is aligned by HighSet, but completely missed by Palpad and star. This exon here may be homologous with some repeat regions, and star and Palpad are unable to align this junction. But because HighSet restricts the alignment to local regions, it can find this junction. So that was a simulation study. We also considered some real data. So our 200 million 100 base pair RNA-Seq data set, we aligned these with the 3 aligners. And because we don't have a ground truth here, we use concordance with ESTs for validation. So then we considered some examples of junctions which can only be detected by a single aligner. These are junctions which could be validated by ESTs, but only detected by a single alignment tool. We see that in real data, there is also this case, as we saw in the simulated data. For instance, here, only Palpad was able to detect the junction. Again, here, Palpad was the only tool able to detect this junction. So it's quite obviously quite actually a true junction. Here, this is a particularly large intron, which is only detected by star. And another junction only detected by star. This is probably due to sequencing errors or some kind or complex region in the genome. And finally, here's a junction only detectable by HighSet. So in conclusion, by considering the merged result of these 3 alignment tools, we're able to get the most comprehensive view of the transcriptome possible. Although RNA-Jam only does a fairly simple merge right now, we are working on more intelligent merging of the tools, even though this simple merge is also very useful in terms of getting good alignments. In terms of other future work that we're working on to improve RNA-Seq results, we're working on incorporating 3rd-generation sequencing into our RNA-Seq workflow, which is able to capture long isoforms and get long-range information between exons. And also, we're working on tools for workflows for gene fusion detection, allele-specific expression, and RNA-Seq variant calling. So that's what I have.
Thank you, John. So before we go into the Q&A session, I wanted to mention the Bina Research Grant Program. If you're an academic and you have a next-generation sequencing project in mind, I would encourage you to apply on our website for our research grant program. On a project basis, we'll give you access to our, thank you. On a project basis, we'll give you access to Bina Ray for secondary analysis, Bina AIM for tertiary analysis, whether you have whole genome sequencing, whole exome sequencing, RNA-Seq, or targeted panels, those are all applicable for your for the research grant program. And then we will support you to present at scientific conferences or user meetings. If you'd like to learn more about this, please just do a Google search for Bina Research Grant or go to this URL listed on the slide. As well, please email me and we can discuss more about details of the grant program. That's anoop.graywall@bina.roche.com. So and then finally, if you'd like to evaluate the RAID solution or the AIM solution, just go to our website, bina.com/testdrive. All right, we're ready for our Q&A. And once again, just a short reminder, hover over the green bar at the top, select Q&A. That'll open up the chat box. Enter your question, and choose to send it to all panelists so that we can all see it and respond appropriately. And then more details about the chat box. But let's get started with some of the questions that are coming in.
Okay, so I'll answer some of these questions here. So I have a question here If the genome of a particular species is non-existent, then is alignment impossible? Well, yes. So the RNA-Seq workflow that we have described, where we align to a reference genome, that would not be possible. However, it is possible to do de novo assembly of the transcriptome using tools such as Transvis and similar tools. And this is currently not supported under Bina Rave, but it's something that we are definitely looking into. So here's another question. Please comment on Sailfish and Callisto. So for those who are unfamiliar, Sailfish and Callisto are quite interesting tools. Instead of doing alignment to a genome, they kind of break the read up into k-mers and do a k-mer match with a reference transcriptome. So these tools are able to quantify the expression of transcripts, but only known transcripts. So they're very fast, and especially Callisto is a very interesting tool, and they're very useful to get especially an initial view of your data. So to see if there's anything interesting or anything wrong with your data. But in order to detect novel isoforms or novel gene expression, then novel genes, then these tools are not appropriate. But if you're only looking at known genes and known transcripts, then definitely these tools are quite useful. And we are looking at integrating some of these into Bina Rave. So I have a question that is asking if the example I gave was 100 base pair reads with 200 million reads. However, in our lab, the RNA-Seq data is 75 base pairs with 30 million reads. Do you think this is a big issue? It depends on what you're trying to detect. If the transcripts that you're interested in are low abundance, then you want higher coverage. But I mean, in general, the amount of data you have is about right to be able to for RNA-Seq. So it's not too big an issue. Yes. So here's a question. Would combining alignments and junctions from Palpad, star, and HighSet increase false positives? It would be best to compare AUC sensitivity, specificity between results from a single software and the Ensemble approach. Yes, I agree that it would increase false alignments. However, in some cases, it's the scientist is interested in discovering some novel splicing events. And because novel splicing events are quite rare in the whole genome sense, that we want to be comprehensive in detecting all possible novel splicing events, and then perhaps do a subsequent study to identify which of these are false and which of these are true. The question, do these workflows enable the detection of expressed genotypes, as you mentioned early on? So I'm guessing this is asking about the concordance between or the relationship between the DNA variants and the RNA-Seq expression. Yes, currently our tools are not able to make this connection, but it's something that we are thinking of integrating into our tertiary analysis. There's a question for isoform detection. Do you recommend single end or paired end reads? I definitely recommend paired end reads because you have a much wider total fragment size, and that's able to link novel splicing junctions together. So that's definitely paired end reads. Many questions I missed? Okay, so there's a question of in what format will the output of RNA-Jam will be? RNA-Jam will output in BAM format, so it basically aligns the input BAM files and then outputs a single BAM file of the 3 aligners. So you'll also annotate within each alignment record which aligners generated that alignment. There's a question does cufflinks and cuffdiff have their own output formats? Yes, they have their own proprietary output formats. The question, can I say star is more sensitive than top hat 2? Yes, it is more sensitive in general because it does find more junctions and more true junctions. However, it misses some junctions which are able to be found by top hat 2. There's a question about what does the QC look like? So, Anoop, are you able to give a quick demo of that?
Sure. All right, we have Rave open, and we're under in the View Job section, we're taking a look at an RNA-Seq project. And underneath, we have the multi-sample statistics coming from FastQC, RCQC, and we're also seeing Palpad statistics.
Okay, sir. Yeah, okay, sure. So there's another question. How are data sets uploaded for RNA-Seq analysis? So, well, it depends on your deployment for Bina Rave. Now, if your deployment is an on-premise deployment, then the data sets will be basically read from shared storage. So any storage system that you have, we connect to your storage system, and then the Rave system will read the data from your own shared storage. For AWS deployments, then it reads from S3, I think. But that question probably is better answered by someone who has done the AWS deployment.
All right, thank you, everyone. If you have any additional questions, please reach out to us, and we can follow up with you afterwards. We'll also be making a recording of this webinar available to you. We'll send you an email about that. And again, thank you very much for attending.